Meiosis is a lot like mitosis. It starts with interphase, but remember, the point of mitosis is to make two identical cells. And in meiosis, what we're trying to do are make four genetically different cells. Because they're destined to be gametes. They're destined to be sperm and egg. Meiosis is a type of cell division in sexually reproducing organisms that reduces the number of chromosomes in gametes, that is, the sex cells, or egg and sperm. In humans, body or somatic cells are diploid, containing two sets of chromosomes, one from each parent. To maintain this state, the egg and sperm that unite during fertilization must be haploid, with a single set of chromosomes. During meiosis, each diploid cell undergoes 2 rounds of division to yield 4 haploid daughter cells, the gametes. Humans have 46 chromosomes in almost every cell, 23 that came from one of our parents, and 23 very similar chromosomes that came from the other one of our parents. We can think of meiosis as a way cells very carefully count and divide their chromosomes so that each gamete, each egg or sperm, has exactly 23 chromosomes. Then, when an egg with 23 chromosomes is fertilized by a sperm with its 23 chromosomes, the resulting fertilized egg has exactly 46 chromosomes. And a new human that grows from that fertilized egg will have 46 chromosomes in all its cells. It is important to have the right number of chromosomes in a cell. If a cell has extra chromosomes or is missing a chromosome, that can have a very substantial impact on how it functions. The gametes, which are essential for sexual reproduction, are produced through meiosis. It can be divided into 9 stages. Here less than 9 stages are shown for convenience, but they will be explained later. The stages of cell division are like mitosis, only difference is that sex cells undergo meiosis twice. So, the stages are divided between the first time the cell divides, which is meiosis 1, and the second time it divides, which is meiosis 2. Let's go through the phases of meiosis 1 first. Although cells do divide, most of the time they are not dividing. The time spent not dividing is called the interphase, or the phase in between divisions. During this phase, chromatin, which is the raw genetic material present in the nucleus, starts to coil up in order to make up the chromosomes before entering prophase. Also, the DNA in the cell is copied, resulting in 2 identical full sets of chromosomes. Outside of the nucleus, centrosomes also start to replicate, each containing a pair of centrioles. These structures are critical for the process of cell division. The next phase in meiosis 1 is prophase 1. First, the centrosomes start to move at the opposite sides of the nucleus. Inside the nucleus, the copied chromosomes condense into X-shaped structures that can be easily seen under a microscope. The meiotic spindle, consisting of microtubules and other proteins, extends across the cell between the centrioles. Each chromosome is composed of 2 sister chromatids containing identical genetic information. The chromosomes pair up so that both copies of chromosome 1 are together, both copies of chromosome 2 are together, and so on. During prophase, either chromosomes undergo what's called synapsis, and so what's happening in chromosome 1 from dad and 1 from mom are coming together, and they're wrapping around each other tightly. Upon closer look, the chromosomes that have come together are swapping genes. In other words, these are pretty much identical except for the changes in the genes. And so, they undergo what's called crossing over. So segments of chromosomes from mom are switching with chromosomes from dad. Now, what's that giving us? It's giving variation. At the end of prophase 1, the membrane around the nucleus in the cell dissolves away, releasing the chromosomes. After this, the next phase of meiosis starts, which is known as the metaphase. In the phase, the chromosome pairs line up next to each other along the center, equator of the cell. The imaginary line where these chromosomes align is also called the metaphase plate. Another thing to note here is that the arrangement of chromosomes determines the degree of genetic variation. If you look at their arrangement here, they could have organized themselves like this: red and yellow ones on the left, and the blue and green ones on the right. But they could have easily been organized in different ways, as shown here. So, they could have been in a different position. So, this would be a totally different orientation of the chromosomes. It also could organize like this, and also like this. And so, what do we have? We have 4 different ways that these chromosomes could orient themselves independently at metaphase 1. What is that giving us? Well, it's giving us variation. In humans, we have 23 chromosomes. And so, there are over 8 million ways that all of those chromosomes could independently orient themselves during metaphase 1. Other than the orientation of chromosomes, in metaphase 1, the meiotic spindle fibers attach to one chromosome of each pair. After this, the next phase starts the anaphase, in which the pair of chromosomes are then pulled apart by the meiotic spindle, which pulls one chromosome to one pole of the cell and the other chromosome to the opposite pole. In meiosis 1, the sister chromatids stay together. This is different from what happens in mitosis and meiosis 2. The chromosomes complete their move to the opposite poles of the cell. In telophase, at each pole of the cell, a full set of chromosomes gather. A membrane forms around each set of chromosomes to create 2 new nuclei. The single cell then pinches in the middle to form 2 separate daughter cells, each containing a full set of chromosomes within a nucleus. This process is known as cytokinesis. From here, meiosis 2 starts, which has the same phases as meiosis 1, but with just a few differences that I'll walk you through. Prophase 2 starts now that there are 2 daughter cells, each with 23 chromosomes, 23 pairs of chromatids. There is no synapsis and crossing over in prophase 2 as compared to prophase 1. In prophase 2, the membrane around the nucleus in each daughter cell dissolves away, releasing the chromosomes. While the centrioles duplicate and the meiotic spindle forms again, in each of the 2 daughter cells, the chromosomes, pair of sister chromatids, line up end to end along the equator of the cell. The centrioles are now at opposite poles in each of the daughter cells. Meiotic spindle fibers at each pole of the cell attach to each of the sister chromatids. After that, an enzyme called the separase acts on the cohesion proteins to disintegrate them, so the sister chromatids can be separated in the next phase. At the stating of anaphase 2, the sister chromatids are then pulled by the action of the meiotic spindle. The separated chromatids are now individual chromosomes and are present at one pole of the cell. The last stage, that is the telophase, following the completion of the move to the opposite poles of the cell. At each pole of the cell, a full set of chromosomes gather. A membrane forms around each set of chromosomes to create 2 new cell nuclei. This is the last phase of meiosis. However, cell division is not complete without another round of cytokinesis. Once cytokinesis is complete, there are 4 granddaughter cells, each with half a set of chromosomes, haploid. In males, these 4 cells are all sperm cells. In females, one of the cells is an egg cell, while the other 3 are polar bodies, small cells that do not develop into eggs. That is all about the process of meiosis, through which sex cells divide and form.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.